And the title of my presentation is brain-PBM, How to Inform Next Steps in Clinical Trials, the case of Major Depressive Disorder. So with that being said, I do have some disclosures. I'm the co-founder of a company in the light space. However, no devices of these companies are presented here, and no trials of this company are also presented here. So I have to tell you that we have been scratching our head, and when I say we, I'm talking about the PBM team here in psychiatry, collaborators at NYU, NKI, Nathan Klein Institute, and companies that have been working with us in recent trials, trying to figure out what to make of the results that we recently had with our trials. And so I knew coming here that there would be a fair amount of nonsense. And I actually, fortunately, took a trip to Mexico with my family. There were a fair amount of flights. I had time to think, and I was trying to figure out what would be the interpretation, the understanding that I would be communicating today. And it wasn't easy. And just last Thursday, I realized that, I probably would have had no clear explanation, and that it was okay to come in with my problems and my questions. So in that spirit, I'm presenting to you today. So let's start with the case of depression. So maybe a little bit of history of our group and our depression program. In 2008, Dr. Iosifescu, that back then was part of the depression program, studied brain metabolism in depression and found that with the MRS spectroscopy and phosphorus spectroscopy, that indeed the depressed brain has a low cellular energy level. And interestingly, the antidepressant treatment could modify that as long as people were indeed responding to the antidepressant medication. You could actually see the indicator of NTP level, nucleotide triphosphate, increasing in this voxel of the depressed brain as response occurred. This was different from non-responders. So that really launched us into this understanding of depression as a metabolic or hypometabolic condition. Other groups have deeply emphasized how the mitochondria then in the depressed subject here in pink differs highly so compared to the healthy subjects. And that's for a number of structural and functional reasons. So there are DNA damages, mutation, polymorphism, abnormal cristae, but also abnormal electron transport chain, ATP production is abnormal. I'm sorry, they are knocking on my door. Come in. Okay. Let me put my mask. My office is also the storage for our laser devices. So, so these abnormalities tend to then produce other changes. Forgive me. Other changes that are related to apoptosis, inflammation, also oxidative stress, and interestingly, antidepressant medication counteract some of these changes. So you can imagine how delighted we were to think about infrared light as an optimal way to target mitochondria, reestablish metabolism, and have a trickle-down effect. The Wellman Center had, moreover, with extensive research conducted by you, showed specific thresholds in cortical neurons in terms of energy deposition or fluence, I will say, at which you could expect ATP changes. Here was 3 joule per square centimeter. There were thresholds also determined for other physiological mitochondrial changes, like changes in mitochondrial membrane potential that occurred with a lower threshold here, 0.3 joule per square centimeter. So with that in mind, we embarked in our first study, which was called ELATED 2. I will say here that this was our first double-blind randomized controlled trial in depression. Just a few notes about how I'm presenting the parameters, because I will follow this structure also with other slides. Here I'm talking about the overall power per device. In some studies like this one, there are 2 devices. So to keep that in mind, when I refer to the irradiance, I will always refer in this slide to the average irradiance. Here, of course, that makes no difference. When I refer to the surface, I refer, if there are 2 devices or multiple sites, I refer to the total treatment surface. Here is the total energy per session. Here I will indicate if there are multiple sites or if it's bilateral or unilateral. And down below, I'll put down the number of sessions and the overall follow-up. So with that in mind, we had actually a very successful study where we showed that the depression scores over time decreased significantly in the treated subjects with at least 10 points decrease in the depression severity, and that was significant compared to the sham. So the effect size also was moderate to high. And so this was our perfect world. We believed, depending where we are in kind of the debate, that in brain-PBM and IR photons reach the target brain region, which was for us the dorsolateral prefrontal cortex, and is typically neuromodulated, in TMS and TDCS, so in other techniques as well of stimulation. Then the photons are absorbed by the mitochondria, lead to metabolic changes that are responsible for neurotherapeutic effects. However, not so simple. And I'll just move on and talk a little more about this low dose. So as we were interested in these thresholds, we were particularly wondering about what energy and what parameters are necessary to shed near-infrared light over the forehead in order to reach this threshold of energy deposition at the target. We embarked in a study in collaboration with Chen Feng at Northeastern University and his group, and we looked at Monte Carlo simulation, Monte Carlo Extreme, looking at mesh models of the brain where we were specifically determining optical coefficients to understand light permeability of different tissues and reconstructing based on the known distribution of this tissue in the brain and, of course, in the extracerebral tissues as well. It's no surprise that we found a logarithmic decay of energy as the thickness of extracerebral tissues increased. This in green and red are our typical case scenarios of aiming through these 2 points, F3, F4, in the EEG point reference and aiming through that to the dorsolateral prefrontal cortex. So what we found is that with parameters similar to the ones that I mentioned, and just roughly interpreting the amount of energy distributed over the known surface, that we could predict barely reaching a 0.3 threshold of energy density or fluence, I should say, at the target in terms of joule per square centimeters. So what that meant was a bit surprising to us. It was that if you think about the median, that there was half of that area that we were targeting that was actually subthreshold. Not only that, but this is a fairly large area. So depending on whether you were in a caudal or rostral part, you could have a different distribution. It's no surprise that thinking of the anatomy that you have gyri and sulci, and sulci that within the same functional area of the brain are of course much more distant. But it was a surprise that we're barely reaching threshold as a median, and a large portion of our target area was therefore subthreshold, and yet we were seeing some benefits from a clinical standpoint at least. We went on with a larger study, this time in collaboration with Dr. Iosifescu, now at NYU. We had a somewhat of fancier device, however, the parameters were supposedly fairly similar, and the target regions are fairly also overlapping. What we found here is that the difference in between the sham here in orange and the NIR treatment, this was a two-phase study, phase 1 and phase 2, but in both phases was really not significant, and the effect size was very small, fairly small. So this was not because the sham was particularly effective. As you see here, there were only a 5-point decrease in the scores for depression severity. So again, we were wondering, okay, this threshold dose or energy deposition that we expect with this range of parameters, is this effective, is this not effective? At the same time, we were conducting a study in healthy subjects for the same low dose. We're wondering specifically whether that same low dose that we used or were using in depressed subjects was indeed engaging brain function. And to do that, we recorded electrical brain oscillations through EEG recording before and after treatment. We use a crossover design, so that means like we were moving from one dose to the other on weekly sessions, and we were also testing sham. This was a single blind study, while all prior studies were double blind studies. So what we found with the exact same dose that we tested in depressed subjects is that there was indeed an effect in amplifying both gamma and beta EEG waves. So this was the before treatment, pre-light, and this was after treatment, and this is already corrected for the effect of sham, as you see here, CW minus sham. So it's clear that there was a gamma and beta enhancement, and now we had this somewhat conflicting data that was showing that this dose, this low dose, indeed engaged in some ways the target area, inducing kind of fast brain oscillations were typically correlated with executive functions or improvement in executive function and attention. And yet this dose in the depressed brain, in the depressed subjects, didn't quite have an equivocal benefit. So we went back to our studies with Northeastern University with the Chen Chen Fang group, and we wonder about the differences in population. Our depressed subjects were more in their mid 40s or early 50s, while our healthy volunteers were typically in their kind of 20s, and you can see that there is a difference in expected thickness of extracerebral tissues. This might be an overestimate because probably people in their 20s are right here, but still we wondered whether that could have an effect. If we think about this level of change in thickness between 8 to 12 millimeter of extracerebral tissues, you would expect at least a half order of magnitude in attenuation. So that could have been an explanation. We don't know. We haven't really tested this paradigm of engagement of brain oscillation in depressed subjects. What we did, however, we tested a PW dose, which was on a duty cycle of 33%, and was therefore 1/3 lower in terms of total energy deposition and also the other parameters. What happened here is that we witnessed a complete loss of the engagement in healthy subjects of our brain oscillations, both for gamma and beta. So with no difference here, you see before and after treatment. This was equivalent to this kind of schematic of predicted deposition. Now the time of irradiance was lower, the total energy was lower, however, we could use this framework to say that according to this model, we could predict maybe 0.1 joule per square centimeter of deposition at target area. So with that in mind, that study maybe had some confirmation of what had been seen at the Wellman Center in neuronal culture, maybe indeed stepping below this threshold of 0.3 joule per square centimeter predicted a target area meant losing neuromodulatory effects. So then what about the depressed brain? Is there any indication that there could be, with a low dose, any engagement of brain function in the depressed brain? We ran a study in collaboration with NYU, and these are our preliminary results just presented at the Society of Biological Psychiatry this year. Again, similar fluence. To note here is that we this time use a laser device. All the data that I presented so far were on LED devices. But otherwise, we tried to reproduce fairly similarly the parameters. And we looked here at the MRI signal. For the first time, we recorded during treatment, not before and after. And we looked specifically at the blood oxygenation dependent signal, so the bold signal, with the goal primarily to have some indication of potential modulation of cerebral blood flow. What we saw here on the left is that for the sham condition, there was no difference whatsoever. This was also a crossover design similarly to the studies on electrophysiology. And on a weekly basis, subjects were presented with different doses. The low dose this time produced a remarkable change. This is a very strong effect in terms of during the stimulation, not before and not even after, but just during the stimulation of the bold signal. So that was not what we expected, but definitely indicated that the low dose could engage brain function even during treatment and in depressed subjects. So I'll say here that not all brain PBM is equal. And with that, I want to transition immediately to the medium dose. Same paradigm. At this time, we had twice as much energy over the same surface. So this was a CW dose. This was, you can imagine, the week before or 2 weeks after. And this time, we saw a remarkable increase in both signal, which is very different from sham, and the opposite of what we saw with low dose. So we had not predicted that either, that there would be this discrepancy in between the low dose and the medium dose in terms of engagement. Again, a robust effect and significant effect. Going back to our prediction model based on the collaboration with Neurophysiological University and the Monte Carlo Extreme, interestingly, when we were computing and finding what would be the likely energy deposition and effluence at target, these were not so different in between the medium dose and the low dose, 0.5 in 1 and 0.3. Of course, 0.5 meant that we were confidently above that threshold that we were considering as a reference point. But noticeably in the cultures, you see a fairly linear effect and a significant effect that is fairly unidirectional in terms of mitochondrial membrane potential, whether you are at 0.3 joule per square centimeter or whether you are above that. So that was another reason of discrepancy that led us to question our overall model. If you think about it in the low dose, we were wondering, okay, how is that was a subthreshold dose or a dose that reaches the threshold, but just as a median, you're gaining an effect, and how comes now that you have discrepant and opposite effect while you're predicting fairly similar energy depositions between the low and the medium dose. So more reason to question our model. Well, just a hypothesis that we are bringing over here is that the bold signal is indeed very much driven by the deoxyhemoglobin, and of course, cerebral blood flow affects the bold signal, and the greater is the cerebral blood flow, the greater is the bold. However, because that means oxygen supply. However, brain activation means oxygen extraction. Possibly, medium dose means more photon than the low dose, more nitric oxide release, more cerebral blood flow, possibly exceeding the brain activity. In the low dose, we saw that there was clearly brain activation. Maybe the effect on cerebral blood flow is not such that to counteract and overcome the extraction of oxygen related to brain activation. That's a hypothesis. At this point, we really don't know what's the best explanation of these discordant findings. I will say that although the study wasn't meant to look at clinical effects because it was only one dose per week and there was only 1 week of time to look at clinical ratings, we did anyway rate our participants for their depression scores. And we saw that despite the discordant biological findings or neurophysiological findings, both the medium and the low dose tended to produce a decrease in depression severity scores compared to the sham. This was scores that were considered before and after the treatment. So then what happens with a high dose? And here's the time of the paradox. The high dose was a PW dose. This was also a 33% duty cycle, like the PW dose that we had used in prior studies. Much more total energy on the same surface. And what we saw is that the high dose produced no effect whatsoever, very similar to sham. This was also unexpected. And if you think, I mentioned there that the average was close to the average irradiance, close to 300. Actually, the exposure was 10 minutes, so the total energy was 4. So based on this correction, the expected deposition for this dose was about 1 joule per square centimeter. So this was closer to the optimal dose that we considered would give us the greatest effect, both biologically and clinically. However, it didn't biologically, and it didn't clinically. I mentioned before that this reading at 1 week are really rough indication of the potential for antidepressant effect, but you can tell that the high dose is closer to sham in terms of changes in depression scores compared to the medium and the low dose. So this was another reason for being surprised and thinking, well, this doesn't really gel well with what we hold as a framework of how transcranial photobiomodulation would work in neuropsychiatry and in depression specifically. So the title of this section is not just local, the whole brain. We did go on and look at the whole brain effect on bold signal. We were expecting that really the changes would occur where we were targeting, because we knew that beyond that, the likelihood of having sufficient deposition would be very low. However, that wasn't the case. We had similar effects in terms of bold signal extended beyond the target area. That also didn't make sense. And yet, here on top, this table is for the frontal bold signal. This is for the global bold signal. And you can see that the effect size is, if anything, larger. We do have a team with various expertises, people who are experts in brain coherence, in networks, in connectivity. So all this element of synchrony in terms of brain activation of different brain areas. However, none of them thought that those elements could be reasonable explanations of what we saw as the light flash through in our treatments and in terms of bold signal and how generalized the effect was. The NIH requires us to conduct MRS spectroscopy and thermometry to find out if there was any warming associated with this treatment. One concern obviously was, could the warming be responsible for any changes that we are documenting potentially in terms of blood flow? And in brief, I'm going to tell you that when post minus pre in terms of temperature were calculated across different doses, if anything, there was a non-significant slight drop in temperature. These were all, of course, measured in the MR, in highly ventilated space. But there was no indication across this treatment of increase in cerebral temperature. And this is the voxel that was close to the target area. So in conclusion, and some of these conclusions might be outside the box, I will say that there are limitations, obviously, in our model. The way we have been estimating what is expected target deposition of the photons really has all to do with understanding tissue properties and understanding that in relation to a given wavelength. There was no information on the effect of laser expressed on LED, no information about high irradiance or even peak irradiance as an even higher irradiance or high frequency of pulses that could affect light penetration. And also in terms of tissue, there was no clear understanding of the healthy versus diseased brain, and how could that potentially affect the permeability to light. In terms of main finding related to the photon deposition at target, I'll summarize one more time that again, the barely threshold, also threshold NIR deposition at target leads to neuromodulation. That was somewhat surprising based on that median and the fact that half of the area was estimated to be subthreshold. Within range NIR, when you think about low and medium dose, how close they were led to opposite effect in terms of neuromodulation. When you think about the bold effects in the MR that I show you. And then close to optimal, expected optimal NIR leads to absence of neuromodulation. That's the high dose that I showed had no clinical effect, no biological effect in our case. And finally, local NIR leads to global brain-wide neuromodulation, which is also counterintuitive given the fact that we know that the photons won't go that far. So what researchers have done when this somewhat conflicting and puzzling result come about, they have put some faith into the systemic effects, which are certainly happening. We know about systemic effects related to anti-inflammatory effect, antioxidant effect. We know that there are free circulating mitochondria that can be modulated. However, they are unlikely to explain what we have seen here related, and this is because of the rapid onset of neuromodulation within minutes, the rapid dissipation of neuromodulation once the treatment was stopped. And other groups have also emphasized that there is laterality of brain-PBM. For instance, Barney Mazur in her studies of TBI, or even Francisco Gonzalez Lima's group have shown some the location of the light source matters. Some speculations here, the distribution of photons over time might be a signal. We have looked at the average fluence at target. So how many photons reach the target in our estimation? And we were content with that. However, you know, really the temporal pattern of photons and how intense the light might be at certain points and might not be at other points might matter and might represent a signal, and maybe the brain responds to that signal. So we definitely need to pay more attention to radiance, peak irradiance, pulse frequency, duty cycle, exposure time, and see how those potentially convey a signal to the brain. Ripple effect, we've focused very much in our model into where does the photon drops and where, so where is that, and the effect should be there. However, what if there were ripple effects, whether those could be related to speckle phenomena or speckle effects, or whether related to biophotons? Is there something else that would explain why we have signals that are more global than local. Finally, again, another limitation is when we consider light penetration and total energy delivered in our model, we thought that that could estimate, with fair certainty what would be the effect in terms of biological and therapeutic effect. And in this model, we haven't really looked at the tissue responsiveness. How does the tissue, how does brain tissue respond? And that could vary based on the type of node, the network, but also whether it's healthy or diseased, young or aging, and many other aspects that could, for instance, affect mitochondrial function if that's really our focus. Like diet, there's a typo here, physical activity, sleep quality, supplements, medications, drugs. So we haven't really thought about much about this tissue responsiveness. With that, I want to thank our team, and there are many, many groups, our closest PBM team, the neuromod, larger neuromod team, who's also informed our study, our MRS team, the group at NYU NKI, the CUNY group that has contributed to the analysis, LightCure as well, who provided us with the devices. This was our team to a recent walk to end Alzheimer. We have studies on Alzheimer as well. And finally, I would like to thank you for listening today, for inviting me. I promise I wouldn't have answers, but just questions, and I think I followed through. Thank you.
Thank you very much, Dr. Cassano. That was a really interesting talk, and certainly very different from at least what a lot of us are working on here at Wellman. So I'd like to open the floor to any questions. And I see Zainab has a question.
Yes, very interesting talk. Many questions, actually, more questions from probably what you were expecting to raise. But one question about the optical properties of brain. Have you looked into differences in optical properties in depressed brain and normal brain? Would that explain the penetration of the light in healthy and depressed subjects? And like you mentioned that there are other drugs that might affect the depression and energy level in mitochondria. Can you comment on, for example, the effect of caffeine, serotonin, and other substances that it's told that they have antidepressant effect? How about supplements such as CoQ10? Those kind of things are expected to raise the energy level. Do you expect them affecting the brain in any way?
Sure. So thank you so much for your question. So in terms of optical properties in the depressed brain, we haven't looked at that. So it's something that would be interesting. One aspect I didn't mention is that there are many potential subtypes of depression, and it might be different to have more of a vascular depression in the aging brain from different subtypes in the young brain, or even a melancholic depression and a typical depression. They have different symptoms. So we don't have an answer to that. I think it's an important aspect to understand if there are different optical properties. In terms of serotonin and caffeine, I can tell you about serotonin. Indeed, those medications that I was referring to from Dan Yosefescu's earlier studies were serotonergic medication. So those were the medications that indeed affected metabolism and metabolic changes. I don't know about their effect on optical properties. The Q10, as you know, certainly affects mitochondrial function. Could potentially amplify the effect of PBM. I recall a recent study that looked at Q10 and PBM augmentation, and it wasn't obvious that there was a synergistic effect.
And Rox, you have a question?
Yeah, great talk. I love it when there's more questions than answers. That's what real science is about, right? It's what you don't know. So first I have a comment and then a question there. You know, this guy, Furchtgott, won the Nobel Prize shared it actually, for discovering nitric oxide. And there is a phenomenon, optical phenomenon called Furchtgott's phenomenon, by which light will immediately dilate vessels. Mechanism not exactly clear. It may be related to release of NO stores from hemoglobin binding. And it's very similar to the photochemistry that we suspect is going on in mitochondria for photobiomodulation, where there's an NO sitting on a regulatory site in the cytochrome C complex. So I think your bold imaging, thank you for doing that, by the way, because it's a quantitative endpoint. You can't, you know, it's not a fuzzy endpoint. You either change the blood oxygenation signal or not. But it may be that it has nothing to do with photobiomodulation at the mitochondrial level. It may be a direct effect on the microvasculature of the brain. And maybe you'll have a comment for me about that. But my question is actually along a similar line. I think in this field, there's a real need for quantitative information about how light affects the brain. And so many of these processes lead to cortical atrophy. So you have natural aging, you have a certain rate of cortical atrophy, and then it's greatly accelerated in the neurodegenerative disorders. With high resolution MRI, it's now possible to look at the cortical thickness at the level of 100 microns or so resolution. Are you aware, Paolo, or maybe you yourself have looked into this, whether photobiomodulation reduces the rate of cortical atrophy in the normal and also the neurodegenerating brain? It just seems like a rock hard endpoint that one could look at. And my understanding is that one of the primary effects, in fact, of photobiomodulation is to stave off apoptosis. So presumably the cortical atrophy is happening because of neuronal dropout from that pathway of cell death. So anyway, but where are we at with that using high-res MRI?
Yes, so we haven't looked at that, but we will be able to look at that. The reason is that so far the MR that we have available is really just related to one treatment, and it's basically 1 week after one treatment. So not so much in terms of treatment effect based on time and based on number of sessions, but we haven't looked at those data either. We will have data now as we will move on from this phase of crossover designs where we compare different doses to selecting a candidate dose and bring it to the next level, which is a randomized trial of 8 weeks in depression. That's the mechanism, by the way, of funding by the NIH. They want one phase, it's called R61, and the second phase, the R33. In those 8 weeks, we probably have more chances to see if there is any effect on cortical thickness. And we are also, as mentioned before, conducting a larger trial that is funded by both the National Institute of Aging and the Alzheimer's Association, multi-site, where we're treating patients with mild cognitive impairment with AD etiology up to mild dementia, and we're treating them for 8 weeks, and we're repeating the MR And so we can look at that in Alzheimer's disease as well.
So in those studies, you'll have a sort of a wash-in period where you document the rate of cortical atrophy, and then you have your intervention. Is that right? How are you going to control for that?
Yes, we have an MR before and MR after the intervention. Is that what you meant?
Well, you're the brain scientist, not me. So it's very dangerous for me to do this. But my understanding from the literature is that if you take two high-res MRs about 6 months apart, you actually get a rate for the cortical atrophy. So how fast is the cortex thinning? And then you would expect, if you had an effective intervention, to change that rate, as opposed to the absolute value of the cortex thickness, which is going to vary from person to person a lot.
Right. So we will not have the opportunity to assess the rate of cortical thinning before they receive treatment. However, because we're comparing sham and treatment, we can see if during the study the rate of thinning is different among the two arms.
Yeah, thank you.
Josh?
Hi, Paolo. Thank you so much for that talk. It's like Zainab was saying, that raised a lot of more questions and answers. And I'll ask you something that's for completely selfish reasons. I'm involved in an effort right now to look at photobiomodulation in the skeletal muscular system. And your talk kind of revived one of my biggest fears about PBM in general, in that I think a lot of our concepts about dosimetry, about where the optimal kind of therapeutic window is, comes from preclinical models where the endpoints are kind of very basic cellular functions. Like in your case, you're looking at membrane changes in membrane potential. Sometimes we look at ATP production or nitric oxide synthesis, things like that. And then I think we make a pretty big leap of faith from that to kind of clinical or in vivo endpoints that involve a lot of different cell types, a lot of different processes, and we just assume that whatever window we see in the cells that changes, ATP production, is the same window that would change how fast you run, for example. And that, for clinical studies, we're in the midst of trying to finalize some parameters for clinical studies, and we have to pick some parameters to use. And it to me, it always feels like, we try to make our best kind of educated judge, as a recommendation, but it always seems to me almost like a flying guess. And I just, I'm sure it looks like you've run across some of these things. I'm just interested to hear your thoughts about that.
Absolutely, I agree with you. It seems like it's really hard to pick the dose. My hope is that we will move past this as we identify ways to personalize the dose. And in kind of brain science, it would mean having sensors that would help us kind of in real time test different doses and see what seems to be most effective. The challenge even with that is that what leads to the best effect in the session is not necessarily what leads to the best effect over time with repeated sessions. We don't have any guarantee that there is a clear, straightforward equation. And also, we don't know necessarily what's the best target engagement, even from a neurophysiological point of view. Is that, brain oscillation? Is it ball signal? Is it something else? We're very much debating what would be the next dose. And in fact, that was the title of my presentation for a good reason.
Yeah, thank you. I'd love to talk to you about this some more in more detail. We'll probably need some alcohol with that, it sounds like. But thank you.
Absolutely.
And perhaps I have one question too, which is a lot of the time when we're thinking about energy deposition onto a tissue and we're thinking about the safety limits associated with that, the calculations that we do are actually based on like the energy deposition into the skin and what the safe limits for that might be. Do you think it's time that we sort of revise these safety limits so that we have sort of tissue specific maximum permissible exposures, etc.? And do you think there's any drive towards doing that at all?
I haven't looked too much into this. Maybe I should have. What I can say is that Forterra and Luis de Taboada have led extensive trials besides their NEST trial, which was the largest in humans, but extensive experimentation in animal models and in neuronal tissue, and they clearly map what is a dose that is safe. And they went on, having an irradiance of 700 milliwatt per square centimeter in their trial for stroke, and they eventually showed that there was although they were shedding light over the entire head, there was no difference in terms of adverse event in between sham and treated. So I think we're still within range, but if you want more detail, definitely contact Luis.
I mean, that's cool. It sounds like there's a whole data set there that people could actually use and draw on, but I don't know if that's publicly available or, yeah, I don't know.
So I have to comment because like I'm the author of launching God knows how many much more powerful devices that are used on skin. And I was a member of the ANSI Z136 standard years ago. That standard is very, very conservative. It's really a standard for environmental sources of light. And if you have a therapeutic benefit, I wouldn't worry about the skin or the skull table or the calvarium, the stuff between the outside and the brain. For example, when we made a diode array source at exactly the same wavelength that's very popular for laser hair removal, we deliver 50 joules per square centimeter in 50 milliseconds, all right? It's 1,000 watts per square centimeter, whopping dose, way over what you're doing, and people do just fine with that. So I think what drives this is really the interest in helping people with these horrible diseases. And I just think you have a lot of room to wiggle regarding the safety. That's not the issue. And the using quantitative human brain endpoints like the stuff you've done with BOLD or EEG changes and cortical thinning, that's going to be the guide star, I believe, to get the dosimetry correct. Look at the response. I love your work, Paolo. We did a study that you participated in here. I don't know if everybody is aware of it, where we took patients with traumatic brain injury and used a similar approach and found by serial MRI that there's no question that after TBI, photobiomodulation affects the course of healing. And in that study, even though it wasn't configured to measure a clinical outcome, there was actually a significant a strong trend. It didn't reach the clinical significance for people having been treated to have better sleep. So sleep disturbance after TBI is a major disorder. That study was underpowered, and I think that we could probably get DoD to support some serious similar studies since they care so much about that disorder.
That'd be very nice.
All right, so that's after 1. So I'd like to give everyone going to give Dr. Cassano a virtual round of applause again. And thank you very much. It was a great talk.
Thank you. Thank you for having me. Take care.